So welcome everyone. Hello on my behalf as well to this FocalPlane special seminar to kind of celebrate one of our Journal of Cell Science special issues that Lucy and I were co-editing. So I'm just gonna share my screen. Hopefully that I can do that. All right, just to say a few words about the Journal of Cell Science, so the journal which is kind of sponsoring this webinar. As you probably know, so Journal of Cell Science is one of the leading publishing venues for any kind of research related to cell biology. And there's a few things that I think make it a very special journal that you should consider to publish your own research in. And the first one is that it's free to publish. So there's many now journals where you have to pay quite a lot of publishing fees, but this is actually free in JCS. One of the other things that is really good to also consider is that we have a very strong commitment in publishing the manuscripts that we revise. So over 95% of the revisions that we receive end up being published and accepted in Journal of Cell Science. We are preprint friendly. And I think, in my opinion, what's even more important is that we are not a for-profit publishing company, but instead, a lot of the profits are then kind of reinvested in the community to organize meetings and seminars and initiatives like FocalPlane. So one of those initiatives that you may or may not be aware of, which started perhaps a year ago, which I think is very exciting, is that so the company of biologists started the Forest of Biologists, which plant a tree for each of the research article and review published by the journal. And if you review for the journals, then some of the kind of time that you dedicate will then be used to protect some ancient woodland in the UK via this initiative. And so finally, I don't want to take time over the science because we have some very exciting talk coming up, but we just want to further highlight another special issue that will be coming up very soon, which is on cilia and flagella. So if you are doing research in any of those areas, then you should consider submit some of your cool papers to Journal of Cell Science. So on that, you can find more out about this particular journal by going on the website. Okay, so that's all for me. So now I think it's time to get started with the first talk of this webinar, and it's my great pleasure to very briefly introduce Hashim Reza, who will be telling us about some of the exciting work that he published in our special issue on using expansion microscopy. So for that, please share your screen and get started whenever you're ready. I can see your screen very nicely.
Great. Yeah, thank you. So good morning, good afternoon, and good evening, everyone who have joined the webinar from across the globe. And I'd like to start by thanking everyone who was involved in selecting our study for today's talk. And I'll be talking about our work wherein we utilized expansion microscopy to study ultrastructural features of human fungal pathogen Candida albicans. The invention of microscope led to the discovery of cell, and over the years, we have seen advancement in microscopy, which has improved the resolution. However, the resolution limit of fluorescence microscopy coupled with small size cells is a two-step limiting factor, and super resolution microscopy has overcome this problem. However, the problem is super resolution microscopy is sophisticated, it's expensive, and that is why it is restricted to a few users. Now, this is where expansion microscopy comes into play, and it has been a great advancement in the field. Expansion microscopy relies on isotropic physical expansion. It improves the resolution by fold expansion factor, and thereby it overcomes the limitation of fluorescence microscopy. This technique is compatible with both chemical and cryogenic fixation protocols, and recently, Vikas et al. From Gautam's group, they have shown how this technique can be scaled to image for over more than 200 eukaryotic samples. And over the past couple of years, expansion microscopy in some of the fungal species has been worked out, which includes Cerevisiae, S. pombe, Aspergillus fumigatus, and Stylogomeates. And now this is restricted because of the complexity of the cell wall, which needs to be digested before expansion. So in this work, we tried expansion microscopy in the human fungal pathogen Candida albicans because of certain features. Candida albicans exist in various morphotypes, which is very important for its virulence. The genome is highly plastic and it can tolerate various ploidy states. And recently, World Health Organization also placed Candida albicans in critical priority group. Now, as a lab wherein we are interested in cell cycle, we were intrigued by the diversity in spindle positioning across fungal species, wherein, for example, Cerevisiae shows spindle positioning at metaphase close to the bud neck region, Candida albicans shows spindle positioning away from the bud neck, and Cryptococcus neoformans shows migration of the metaphase spindle completely to the daughter cell. Now, one of the important players in spindle positioning is spindle pole body, also known as microtubule organizing centers, wherein it nucleates cytoplasmic or astromicrotubules, which together with various motors and maps and interaction with the cortex plays an important role in spindle positioning. And this is very important for organisms which undergoes asymmetric cell division. However, very little information is available on SPB organization, assembly, and inheritance in Candida albicans. So we optimized Candida albicans extraction protocol, wherein we optimized cell wall digestion, timing, and conditions. This was followed by anchoring of the samples, gelation, and finally staining and expansion. And we were able to expand Candida albicans cells. And here, this is NHS Aster labeled cells. This is pre-expansion cell, and this is post-expansion cell. And with the help of NHS Aster, which stains the proteome of the cell, one can study protein landscape of the cell. And this is where we saw that you can study various organelle structures upon NHS Aster staining. And the first thing what we did was we looked at expansion fold by looking at diameter of non-expanded and expanded cells. And we were able to show that Candida albicans can be expanded by around 4.24 fold, which is similar to what has been reported by for Saccharomyces cerevisiae and S. bombay. So we looked at various subcellular structure at different stages of cell cycle, starting from unbudded, metaphase, early anaphase, and late anaphase. And we saw that there are tubular mitochondria-like structures at all cell cycle stages, which is highlighted by black arrowhead. N represents the nucleus. The yellow arrowhead represents the nucleolus, which is darkly stained by NHS staining. The blue arrowhead represents spindle pole bodies, which is again proteinaceous structure, and it is also stained darkly by NHS staining, and it is present opposed to nucleolus. So we could observe all these structures at different stages of cell cycle, and finally we were able to also see mitotic bridge at late anaphase stage. So the first thing what we did was we validated these tubular structures to be mitochondria by carrying out post-staining with body PTR. Body PTR stains lipids within the cell and are known to stain Golgi bodies and mitochondria. And here we see nice staining of cell membrane and nuclear membrane. And this post-staining with NHS Aster suggested these structures to be mitochondria, and this we see at all stages of cell cycle. As I mentioned earlier, that the nucleolus within the nucleus is stained more with NHS Aster. This we validated by doing chromatic staining using Hoechst, wherein nucleolus is not stained by Hoechst, and this enables an identification of the nucleolus within the cell. We also validated these structures to be nucleolus by carrying out NT-NOP1 staining. NOP1 is a protein which localizes to the nucleolus, and it is important for 18-RS RNA processing. And when we did co-localization for NOP1 with respect to NHS Aster, we see that the darkly stained regions by NHS Aster nicely co-localizes with that of the NOP1. One important feature which we observed by doing NT-NOP1 staining was there was differential staining of the protein, which suggests that protein density of NOP1 within the nucleolus is also different. Now coming to SPB organization. SPB organization in Saccharomyces cerevisiae is divided into three different layers, which is outer plaque, center plaque, and inner plaque. Outer plaque and inner plaque, they are important for nucleation of astromicrotubules and nuclear microtubules respectively. And the SPC 110 is one of the proteins which is present at the inner plaque. So we tagged SPC 110 with GFP, and we validated the NHS stained regions, which was opposite to nucleolus as SPPs or not. So for this, we did anti-GFP staining with for SPC 110, and co-localization studies further validated these structures to be SPBs in albicans. Now, we also noticed that there was differential staining of SPBs with both NHS ester and SPC 110, suggesting that there is asymmetric distribution of proteins between the two SPBs. And we looked at quantification. We quantified the levels of SPC 110 between the old and new SPB, and we see that there is 1.6-fold reduction in signals coming from new SPB as opposed to that of the old SPB. And also, we see that there is no difference in terms of quantification when you do quantification for SPC 110 and NHS ester, which suggests the utility of NHS ester staining in studying asymmetric distribution of proteins within the cell. And asymmetry of SPB during SPB duplication and migration is pretty well worked out in Saccharomyces cerevisiae. We also validated these structures to be microtubule nucleating center by tagging top2 with GFP, and we carried out immunosting using anti-GFP antibodies, and we were able to label the mitotic spindle at various stages of cell cycle, which has been shown over here. And when we did this analysis, we also found that the astral microtubules were probably longer in Candida albicans, which is highlighted over here by blue arrowheads. So we quantified the length of astral microtubule distribution, both in expanded and non-expanded cells. And we found that the astral microtubules were in the range of 2.1 to 2.6 micron, which is like longer as what has been reported by Saccharomyces cerevisiae, which is wherein the length of astral microtubule is less than 1.5 micron. Now, one of the applications of expansion microscopy is decrowding of proteins and cellular space, and this enables the spatial resolution of the proteins. So we wanted to ask if we can resolve the outer and inner block in Candida albicans, and for this we resorted to one of the proteins, which is TUF4. TUF4 is present at both outer block and inner block, and it nucleates nuclear microtubules and cytoplasmic microtubules. This is metaphase cells of Candida albicans, wherein we see two SPB dots, which is marked by blue and yellow arrowheads. And this is NHS ester staining together with Hook staining. And when we carried out anti-GFP staining for TUF4, at low exposure, we were able to resolve two TUF4 dots at the old SPB, and at higher exposure, we were able to see another two dots corresponding to inner and outer block. Now, this observation also suggested that there was no significant difference in TUF4 signals between the old SPB, while this difference was significant or apparent at the new SPB in albicans. We also quantified the distance between inner and outer block, and we found that the distance is around 100 nanometer, and this is smaller as compared to cerevisiae, wherein the distance is reported to be between 140 to 150 nanometer. Now, while we were doing this analysis, we also found a peculiar arrangement of SPB in Candida albicans at metaphase stage, wherein we see that the SPBs are placed close to each other. And this we call as side-by-side arrangement in Candida albicans, as opposed to pole-to-pole arrangement of SPB in Saccharomyces cerevisiae at metaphase. And this indirectly also suggests that the metaphase spindle in Candida albicans is smaller as compared to that of cerevisiae. So this we validated in non-expanded cells, wherein we had tibble intact, and one of the SPB markers, for example, STC42 for cerevisiae and TUF4 for albicans were tagged. And we did Hook staining to mark the chromatin, and we divided the chromatin into two halves, which is minor segment and major segment. And we show that the percentage area covered by minor segment in Candida albicans is around 23% as opposed to 32%, which again hints towards smaller spindle in Candida albicans. Finally, we tried our expansion protocol with few modifications in 6 of the human fungal pathogens which we work in the lab. And these belong to ascomycete and basidiomycete. And we show that at least 3 of the fungal species, which includes Candida parapsilosis, dubliniensis, and N. glabratus, could be expanded by 4-fold, and others could be expanded in the range of 2.5 to 3-fold. We were also able to observe various subcellular features, which included looking at mitochondria, nucleus, spindle pole bodies, and vacuoles. Uh, uh, and finally, we asked what is the status of SPB arrangement in two of the fungal species, N. glabratus, which belongs to whole genome duplication clade, and C. dubliniensis, which belongs to CUG SER1 clade. And we see that both of these fungal species show side-by-side arrangement of the SPBs, which suggests that probably this could be unifying features across fungal species, which needs to be validated further. Today what I've discussed is like are certain unique features in Candida albicans. When we compare with respect to that of the Cerevisiae, we see that the TUF4 intensity difference during metaphase at the old SPB is not so evident in albicans as opposed to being evident in Cerevisiae. The astral microtubule are longer in albicans and the metaphase spindles are shorter. And in terms of SPB arrangement, we have side-by-side arrangement for albicans as opposed to pole-to-pole arrangement for Cerevisiae. And the TUF4 distance or the distance between inner and outer block is around 100 nanometer as opposed to 144 nanometer, which suggests that there is a difference in SPB organization in both the fungal species. And with this, I'd like to acknowledge Gautam for supporting my application, welcoming me in the lab, and giving me free hand in trying out all the antibodies and stains in the lab. Hiril for teaching expansion microscopy, and also in the optimization, she helped a lot. Professor Kaustubh Sanyal at JCS Bangalore for all the support and guidance during the work. And my co-authors from the lab, Rohit and Srijana, who have been very helpful in finishing this work. And funding agencies, EMBL Corporate Partnership Fellowship Program, IMBO Scientific Exchange Grant, and Department of Biotechnology for the fellowship. Thank you for your attention and time, and I'll be happy to take questions.
All right, thank you very much for this great talk. So to the attendees, please pose a question in the question and answer box. If we don't have time to answer your question, I'm sure that Hashim will be answering the question also in the box during the remainder of the webinar. So there's no question now, but I think we have time for a very quick live question. So maybe I can ask one.
Yeah, sure.
So I find it really interesting that you've tried now expansion microscopy in a lot of different species. Yeah. And then also how you also nicely highlight that the output that you get is quite different. And then you also mentioned that you had to tune a little bit the protocol. So could you tell us maybe a little bit more about that technical side of things? On how much optimization do you actually need to do every time you kind of start working with a new type of sample or species with expansion microscopy?
Yeah, so for few of the fungal species wherein we could achieve fourfold expansion, we had to optimize enzyme types. We tried lyticase, we tried zymolase, and also the incubation time and temperature varies for these organisms. So that's where in terms of technical like aspect comes into play. But for other organisms like Cryptococcus neoformans, wherein we know that the cell wall is quite thick, we could achieve maximum of 2.4 fold expansion. We tried Triton X treatment prior to digesting the cell wall. We also tried a cocktail of various enzymes, including lysine enzyme, which is now not available anymore. But yeah, so these are the conditions which we have to try. And yeah, currently we are also looking at various cocktails which you can try for some of these organisms to fully expand and study some of the things which we are planning to.
All right, thank you so much. Lucy has a hand raised as well.
I do. That was a really cool talk. Thanks, Hashim. I was just wondering if you think that the expansion is linear, and if it's not, do you think it matters?
Yeah. So you're talking about linearity with respect to resolution also when you talk about.
Yeah, just so whether across the whole cell, whether the expansion is linear, and therefore whether things end up in the same position relative to other.
Yeah, that's very important. So that's why we talk about always like isotropic expansion of the sample. And this was one of the questions by reviewer also, wherein we had to show whether the expansion is isotropic or not, because it will change the analysis, whatever we are doing in terms of looking at distribution between inner and outer block. So yeah, it plays an important role, whether if you have isotopic expansion versus not.
All right, thank you so much. So I think we should move on to the next speaker. But if you have any additional questions, please put them in the Q&A box. And I guess I will now hand over to Lucy, who will introduce the next speaker.
Brilliant. Thanks very much, Guillaume. So it's my pleasure to introduce Melanie Rug from the Australian National University. She's an associate professor there and director of the Centre for Advanced Microscopy. And she's a cell biologist and parasitologist who uses a whole range of microscopy techniques with a particular focus on correlative applications. And today, Melanie is going to be talking about lipid droplet dynamics in P. falciparum. So over to you, Melanie.
Thanks very much for the introduction, Lucy, and thanks for inviting me to give a talk at this fabulous event here. So just a brief, a little background on why we should actually care about malaria or about this parasite that I'm going to talk to you about, and the lipid droplet dynamics within Plasmodium falciparum. So despite our concerted efforts to throw multi-pronged approaches at the malaria parasite in order to eradicate this devastating disease, this tiny little organism is still causing health and socioeconomic havoc around the world. So the WHO tells us that in 2022, there were an estimated 250 million malaria cases still in the world and over about 600,000 malaria deaths. And those are mainly children under the age of 5. And this is mainly due to the inaccessibility of people to drugs that we have available or the parasite's resistance to the current frontline drugs. So if we want to come up with new pathways to combat this disease, we need to first understand much more about the biology of the parasite and its interactions with the host. This next slide is actually just going to remind you of the very complex life cycle of Plasmodium falciparum, which is the species that I'm going to talk about today, which is the most deadly one of the human Plasmodium parasites. And so we look at the life cycle and we can see that the parasite has to transition between two intricately different hosts. And one of them is, of course, the mosquito, and the other one is the human host. And to make you familiar with the terminology during the talk, I'll just briefly talk about this asexual blood life cycle. Where the parasite, as a tiny little merozoite, which is about 1 micron in length, invades a human erythrocyte, and then it goes through a ring stage, a trophozoite stage, and then a schizon stage. And in this very late stage, the parasite goes through an asexual multiplication step that is called schizogony, during which it eventually segments into about 8 to 32 daughter merozoites, and they come out of the red blood cell, and then they start the life cycle, the asexual life cycle again. In order to survive this massive shift between vectors and so on, vector and host, the parasite actually needs to grow and multiply. And of course, to sustain those growth and multiplication, they have to acquire a significant number of essential biomolecules. And amongst these, of course, lipids are key molecules, key species that the parasite can either scavenge from the host environment, or it can actually synthesize de novo in a relict plastid, the so-called apicoplast, which you can see at the top right here. Our previous studies have shown, the biochemical studies have shown a drastic change in lipid composition between the uninfected red blood cell on the left and infected red blood cell, which you can see here. And if we look in particular at neutral lipid species and look at diacylglycerol and triacylglycerol, you can see a massive difference between infected red blood cells compared to the uninfected red blood cell. Since those neutral lipids serve a crucial crucial repositories of energy and building material in all eukaryotic cells, we hypothesized that the remarkable enrichment of DAGs and TAGs in P. falciparum might be reflected in the dynamics of the major neutral lipid storage organelles, which are lipid droplets. And here in this cartoon on the right, you can actually see the distinct structure of those lipid droplets, which consists of a hydrophobic core composed of neutral lipids surrounded by a monolayer of phospholipids, which of course contains proteins for the function of these lipid droplets. In order to visualize the fate of the lipid droplets in the asexual life cycle stages of the parasite, we made use of the fluorescent dye Nile red, which is traditionally used as a neutral lipid dye imaged with at an excitation emission spectrum of about 559 nanometers and 636 nanometers, which is depicted up here. And under these conditions, we see a significant increase in neutral lipids throughout the stages from trophozoites to mid-schizons, and then also to late schizons. And we look at this in the quantified table here, or in this graph. We can see exactly that reflected from the trophozoite to the schizon stage. We see an enormous increase here. Whereas if we look at those live or images here, we can actually discern something in the form of lipid droplets from the trophocytes to the mid-schizons. But then in the late schizon stage, the pattern actually looks quite different. And that actually prompted us to actually find a way to probably look at this a little bit differently. So we, in this graph here, or in these images and graph, I show you how we try to make use of the properties of Nile red to actually tease apart the different species of the lipids that we see in our images. And so we made use of the spectral properties of Nile red, displaying altered sensitivity and a spectral shift depending on the lipid environment, using a split emission profile to tackle this idea. And here in the excitation emission profile of about 506 nanometers, excitation 550, emission excitation in yellow. This allows for the observation of mainly non-polar head groups representing the neutral lipids. While if we actually use this different excitation emission profile in magenta, we look at the higher specificity for polar head groups, which would then represent, for example, the phospholipids. And if we look at the quant graph here, we observe that neutral lipid-rich punctae from the trough stage towards increasing, were onwards increasing in number and size, depending on our, or reflecting our integral intensity graph. However, in the schizon stages, you can see that the neutral lipid components decreased back to the trophozoite stage. So this is quite different to what we saw with the traditional use of the spectral capabilities of Nile red, where we saw an increase into the schizon stage. So in order to increase the spatial resolution and to eliminate the discrepancies that we saw in these two experiments, we used an auto-staining method to ultrastructurally examine these parasitized red blood cells in their intra-erythrocytic life cycle stage. And a correlation between the electron density of lipid droplets and neutral lipids content was already demonstrated in previous work in various cell types, where the stains preferentially bind to unsaturated bonds in fatty acids. And what we observed here, the features with high electron density material throughout the life cycle in the 2D, depicted by yellow arrows, are reminiscent of lipid droplets. In order to get a handle on number and size of those lipid droplets, though, which is obviously limited in the 2D imaging, we employed focused ion beam SEM volume imaging. And you can see in orange, those are the lipid droplets that we observed in the cells depicted at the top. We then first quantified the numbers of lipid droplets in the different stages, supporting our light microscopy data, as you can see in this graph, that the neutral lipid enriched lipid droplet number increases from the trophozoites to the mid-Chison stage, and we saw a drop in the segmental stages. And this was even more pronounced when we actually measured the volume of the lipid droplets, and we found that the relative lipid droplet volume per parasite area increased almost tenfold in the mid-Chison stages when compared to the lipid droplet volumes in the trophozoite stages. And we saw a sharp decrease again at late Chison stages, which again corroborated our split emission confocal findings. So from then, we actually wanted to know what is actually the role of these lipid droplets in the asexual life cycle stages of Plasmodium falciparum. And we know from previous studies, biochemical studies, that tags are the major neutral lipid class in lipid droplets. Hence, we hypothesized that tag hydrolysis might be crucial for merozoite formation. Remember those little tiny parasites that form within the Chison stage, which could then explain the reduction of lipid droplet number and volume in segmenting Chisons. And the tag hydrolysis is a universal lipid mechanism. Sorry, I'll swap back to that one. Whereby the tags are broken down into glycerol, as you can see on the left here, glycerol and fatty acids, and that is catalyzed by lipases. And we also know that one of the crucial enzymes involved in tag biosynthesis is diacylglycerol acyl transferase, or DGAT, catalyzing the synthesis of tags. In order to test our hypothesis now and examine the dynamics of neutral lipid synthesis and hydrolysis, we used two commercially available inhibitors of either of these two pathways. And as you can see here, we've got orlistat, which is an anti-obesity drug, which irreversibly inhibits the lipase responsible for the breakdown of tags. And when orlistat was added between 8 and 32 hours after invasion of the parasite of the new red blood cells, the completion of the merozoite formation was hindered to a large extent, with only about 20% of the parasites going through to the next cycle compared to the control cells. So in our split, so we used our split emission experiment again, and we observed a much higher number of neutral lipid containing lipid droplets in control cells, indicating that the hydrolysis of the tag was hindered, preventing the access of fatty acid building blocks for membrane formation. Of course, this is crucial for the segmentation stage of the parasite and when the daughter merozoites are formed. So when the drug was added at a later stage in the life cycle, so down here at 44 hours, we can then see that no lipid droplet accumulation was observed. And we can see that the parasites here go through normally because they have actually been able to use the tag in this cycle. These results support the hypothesis that tags are consumed from the lipid droplets in support of the schizogony of the parasite, and that the fatty acids that are released from the LDs are essential for merozoite development. And now in the next experiment, we checked for the role of tag biosynthesis and storage in lipid droplets in the asexual life cycle. And we used an inhibitor called T863, which is a potent and selective inhibitor of DGATs, DGAT1 in particular in human cells, and which prevents DGAT-mediated tag formation in cells. So when we added those to ring stage parasites, T863 arrested the parasite development. However, when it was added later in the life cycle stage, the parasites seemed to grow slower than the control cells, but they could get through. So we look at our split emission experiment again, and in contrast to the treatment with orlistat, no lipid droplets were accumulating when the drug was added at the early stages, but some very small lipid droplets were added at the trophozoite stage. So this suggests that the inhibitory effect of T863 in comparison to orlistat prevents LD formation when added at an early stage. When the LDs are already present at later stages, the parasites can develop relatively normally. So in summary, what I hope I could convince you of was our quantitative split emission analysis in combination with our FibSem volume imaging revealed spatiotemporal LD dynamics during the asexual replication cycle of the malaria parasite in human erythrocytes. And then in addition, that our inhibitor experiments demonstrated functional duality of the LDs during the asexual developmental stages of P. falciparum. And in addition, we provided evidence for the fundamental importance of tag metabolism for parasite survival, since the interference with tag synthesis, storage, or hydrolysis resulted in detrimental effects for the parasites. And overall, we're really only at the start of understanding LD biology in P. falciparum. But I hope I have convinced you that there's quite an important role potentially for finding new avenues to combat malaria. And last but not least, I'd like to thank everyone who was involved in this work, especially our PhD student who was shared between the Meyer Lab at Research School of Biology and my own lab, Joanne Lee, who unfortunately couldn't be here today. So I'm standing in for her and showing her brilliant part of work that we've published, obviously, in this special feature. And I'd also like to thank our collaborators, Giel van Doren and Humboldt University, Kai Matuszewski, and of course our funding bodies, without whom all of this wouldn't be possible. And thanks for your attention.
Wonderful. Thanks very much, Melanie. It's a very elegant study. If you're in the audience, please do put your questions into the box if you have one. And I'm just going to go ahead and ask Melanie. So the split emission is really beautiful. I was just wondering if it's already been used in mammalian cells, and also to what resolution you can push it. So could you use it to look at neutral lipids within membranes surrounding organelles as well?
That's a very good question. So yes, these studies are actually basically from the 80s. So we've found a few papers where this has been done in mammalian cells before. So this is what prompted us to actually try it in Plasmodium, and it worked beautifully on the lipid droplets. But yes, we do have collaborators as well who want to actually see changes in drug assays, or looking at adding certain drugs and then seeing whether the red blood cell membrane polarity changes upon addition of different inhibitors. But we're really only at the start of identifying how well we can resolve those, because of course there's all sorts of different complexity involved with those types of experiments, since the red blood cells that we're using obviously are from patients, so there would be different ages, and I think not only different lipid environments, but also different lipid compositions that are changing with the age of a red blood cell, for example, might actually be complicating those studies. But let's see how we go. with figuring those out in the next few years.
Very exciting. So we don't have any questions at the moment, and we should probably move on, but please do pop your questions into the box for Hashim and Melanie, and they can be answered there. We can type in. So thank you again, and we're going to move on to our last speaker, who is Vikas Tillu. Vikas is a postdoctoral researcher in the lab of Nicholas Ariotti at the Institute for Molecular Bioscience at the University of Queensland, and he investigates in situ receptor trafficking analysis using cryo-electron tomography. So, Vikas, if you could share your screen. And that looks great.
Yep.
Handing over to you.
Yep. Thanks for introduction, Lucy, and thanks for giving me an opportunity to speak in this forum. So today I will present, just a second. Okay, so today I will present work that was recently published in Journal of Cell Science titled Precision In Situ Cryogenic Correlative Light and Electron Microscopy of Optogenetically Positioned Organelles. I would like to start with acknowledgements. I would like to acknowledge Nick Ariotti and Rob Parton for their constant support and funding on this project. I would like to acknowledge the co-first authors of this study, James Ray, for cell line characterization, and Gregory Redpath for sharing his expertise in optogenetics field. I would also like to thank all past and present members of Ariotti and Parton Group, as well as University of New South Wales Electron Microscopy Unit, Jemfang and the University of Queensland and Institute for Molecular Bioscience for infrastructure support. So this slide highlights the structural biology techniques that are used to address sample diversity across the range, starting from molecules, from particles, all the way to cells and tissues. So Nick Ariotti's lab at the University of Queensland focuses on in situ structural biology of cells. So we mainly look at cells using cryo-electron microscopy and specifically using cryo-electron tomography. So this slide shows a typical electron micrograph, to a transmission electron micrograph of cell in a frozen state on the right. And on the left, you can see a typical electron microscopy grid that consists of a gold or copper bars, as shown here in yellow, and a carbon film is laid on top of it, which contains this characteristic holes on which you can perform transmission electron microscopy. And on the right, you can see a cell that is plated in a frozen state on such electron microscopy substrate. So marked here in the red region is apparent boundary of a cell. And you can see that all the regions inside this boundary are dark, highly electron dense, and not readily accessible for transmission electron microscopy. In order to study the organelles that are present within this region, such as nucleus, perinuclear region, ER, mitochondria, endolysosomal organelles, you have to use a suite of techniques which starts with vitrification or freezing of your cells in live unfixed state, which is followed by correlative cryofluorescence microscopy to find the region of your interest, which is followed by a highly specialized technique, which is called as cryofocus ion beam milling. Essentially, what this technique does is it erases the regions of cells that you don't want to image. And this is typically followed by cryo-electron microscopy or cryo-electron tomography, which is followed by subtomogram averaging and other techniques to yield the structures of your protein or organelle of your interest. So we wanted to avoid this big workflow. So we started looking at cells on this type of grid. And what you can see here, I have marked previously a red boundary that looks like a boundary of cell, but actually the boundary of cell is marked here in a green. And as you can see, the region between the red and the green boundary is quite electron transparent, less dense, and easily accessible for transmission electron microscopy. So first we started looking at variety of cells using transmission electron microscopy to look at any of these cells are thin enough for such type of imaging. So we looked at BHK, MEF, HeLa, MDCK, U2OS, and PTK2. And what we found was U2OS to certain extent, and mostly PTK2 cell line, that is Portruc kidney epithelial cell line, is very thin on plastic or glass surface. So we started characterizing this PTK2 cell line for this paper. And so first what we did was we just plated these PTK2 cells on electron microscopy substrate as shown here on the left-hand side. And we stained cells for DAPI, phalloidin for actin network, and paxillin for focal adhesion. And what we observed was cells actually don't spread that much. So what we did was we added a small amount of amorphous carbon coat on top of the carbon film that is already present there. And what we observed was massive change in actin cytoskeletal network as well as focal adhesion. So these results highlights that the substrate mechanical properties alter cellular spread as well as cellular distribution. So next, what we wanted to look at was the substrate stiffness after addition of carbon layer. So for this, we defined three regions on electron microscopy substrate, region 1, 2, and 3. In interest of time, I will only share one and two region results, and we perform atomic force microscopy to get the Young modulus measurements for each region. So first here shown is the Young modulus of electron microscopy substrate without addition of any carbon film, without addition of any exogenous carbon, and after addition of 5 nanometer and 12.5 nanometer. As you can see, there was massive change in Young modulus which suggests that there is increase in stiffness of the substrate after addition of carbon, amorphous carbon. We also observe similar trend after in the region that is on the gold bar. But the main results that are important here are the region 1 that shows massive changes in the stiffness of the substrate that eventually translates to the cell spread and the distribution on electron microscopy grid. So this is sort of summary of this PTK2 cell line that shows that these cells, they have this perinuclear region which contains nucleus and all other organelles that are not accessible for TEM imaging, whereas these cells also spread very thin on electron microscopy substrate that can be easily accessible using TEM imaging. And this is a serial block phase imaging of the PTK2 cell line that shows this nicely thin spread cell line along the electron microscopy grid. And this is a rendering of the same data that shows these imaging regions that are easily accessible for cryotransmission electron microscopy. So this is the overall workflow of this method, wherein you first carbon coat your grids. It can be 5 nanometer or 12 nanometer carbon coat, and then just sterilize your UV grids to avoid any contamination, followed by cell seeding. Perform your treatment, any transfection or staining that you need to do, and then straight away plunge freeze your cells. This is followed by confocal microscopy to target the regions of your interest, followed by cryotransmission electron microscopy to find the regions that were already imaged in cryoconfocal microscopy. This is followed by Cryo-CLEM registration and then tomography, which is eventually followed by high-resolution fitting of fluorescence and cryo-TEM data. So far I have mentioned you about imaging of the organelles that are present within this region. But what about organelles that are localized to perinuclear regions, such as endolysosomes, mitochondria, or ER? So to image these organelles, we use optogenetics approach, in which for this paper, we have shown example of a cryo-CLEM module that interacts after addition of blue light stimulation. So in this method, we have attached CLEM module to KIF-A motor that shows anterograde transport along with microtubules. And we have attached Cryo-2 module to LAMP1 lysosomal marker. So after blue light stimulation, the LAMP1 coated lysosome can be moved towards cell periphery for imaging. And this is a light microscopy imaging on glass cover slip that shows after light stimulation, the LAMP1 positive vesicles can be seen at the cell periphery. So this is an electron microscopy, cryo-electron microscopy experiment of the same cell line expressing LAMP1 hemichere and KIF-A. This is an area of cell in frozen state where cryoconfocal microscopy is performed. And this is TEM image of the same area of the cell shown here. And this is an overlay of the cryoconfocal microscopy and cryo-TEM microscopy. And these are the three regions that are marked for imaging at high resolution that contains LAMP1 positive lysosomes. And these are the snapshot of cryotomography imaging. On the left-hand side, you can see the 3D projection of the cryotomogram. And on the right, you can see overlay of the fluorescence that nicely matches with the lysosomal vesicles that are shown here. So now in next lab, we are applying these methods to study in situ structural analysis of EGF receptor sorting. So in this example, I'm showing you PTK2 cells that are expressing GFP RAF5A and the EGF Alexa 647 uptake that is performed in PTK2 cells. So this is a cryotomogram of the area that was stained for RAF5A and EGF uptake. And on the right, you can see overlay of the 3D projection of the left-hand side and the overlay of the fluorescence. And you can see the squares marked in black and the red. The square marked in black are the overlay of the green and the red signal, whereas on the right, it's just the EGF signal. And on the left, you can see a vesicle that contains both signal, and you can see nice EGF receptors that are lying inside the endosome shown here. Whereas on the right, you can see vesicle marked in white that contains only EGF that is also marked with EGF receptor. So in next lab, we are applying these methods to study in situ trafficking of EGF receptor and few other receptors. Thank you.
Wonderful. Thanks very much, Vikas. That's very cool. Again, if you'd like to put questions into the Q&A, please do. But I wanted to ask you a bit of a philosophical question. So I was wondering, when you're moving organelles optogenetically out to the periphery of the cell, how long do you think they can stay there before you kind of effectively change their content and their identity and their interactions with other organelles?
So if you keep those organelles long enough, they will eventually show a change in their morphology and their interaction. But the methods that we use are 20 to 30 seconds of stimulation, which is followed by immediately plunge freezing. So cells are actually frozen in that state. So there's no time for rearrangement. So we're actually catching the snapshot that happens immediately after relocation of those organelles. So I think we are avoiding that stage.
And do you think you might get any membrane damage during the process?
I don't think so. And for our work, we are looking at the receptors that are protected inside organelle. So for receptors that are outside the organelle, that might be an issue to some extent. But in our case, yeah, I think we are okay.
A very neat way to access the perinuclear region, where we all know there's all sorts of exciting stuff happening, but has been a bit inaccessible to in situ structural biology so far. So very neat technique. So we don't have any questions, but please do follow up with our speakers if you have questions afterwards. I'd just like to thank all of our speakers again, Hashim, Melanie and Vikas, for their wonderful talks, and to remind you that you can access their full papers in the special issue, along with all of the other papers focused on imaging in the special issue JCS. And I'd also like to thank Guillaume for, it was a fun experience co-editing, and Seema and the team at JCS. And of course, thank Helen for organising today's webinar with FocalPlane, which was all very easy and efficient. So with that, unless Helen, do you have anything you wanted to add?
No, I guess just the same, thanking all the speakers and both you and Guillaume for your wonderful job with the special issue. I think it's been it's a fantastic issue. So I really advise everyone to go check it out. So yeah, thanks for all your work.
Wonderful. And wishing everybody a wonderful day, no matter whether it's morning, lunchtime, afternoon or evening. So we'll see you again. Thank you.
Thank you, everyone. Bye-bye. Have a nice day.
Bye, you too.
Bye.
Thanks.